Hi everybody, we're going to be talking about cellular respiration today. Cellular respiration is the process whereby cells harvest energy to make ATP. ATP is our cellular energy. Cells use it for all the processes they complete to maintain life. We're going to oxidize glucose and transfer the electrons from the bonds that are broken to oxygen. We do this in a series of steps. We can break the steps down by where they are located in the cell. The first phase is glycolysis. This happens in the cytoplasm. The second phase is pyruvate oxidation and the citric acid cycle. That happens in the matrix of the mitochondria. And the third phase is oxidative phosphorylation. That happens across the inner membrane of the mitochondria. There are two mechanisms for making ATP. Both of them involve adding a phosphate to ADP to create ATP. Substrate level phosphorylation involves transferring a phosphate from a substrate onto ADP. This makes ATP. We see this type of ATP production in the first two phases of cellular respiration, glycolysis and the citric acid cycle. Oxidative phosphorylation is the last phase of cellular respiration and utilizes the electron transport chain to create potential energy in a gradient, which can then be harvested to make ATP. This is much more efficient and makes much more ATP than substrate level phosphorylation. 90% of the ATP produced during cellular respiration is made in that third phase. Here's an overview of cellular respiration. Glycolysis is our first step, and this happens in the cytosol, and it does not require oxygen. Therefore, all organisms are capable of glycolysis. Glucose is initially broken down into pyruvate. Some electrons are transferred to NAD, reducing it to NADH. The second phase of cellular respiration is pyruvate oxidation and the citric acid cycle. This happens in the matrix of the mitochondria, which is the innermost part. Glucose becomes completely oxidized during this stage. All of the carbons are released as CO2, or carbon dioxide. During this process, more electrons are used to reduce NAD and FAD to NADH and FADH2. This step does require oxygen, as it has to combine with the carbons to form CO2. The last step of cellular respiration is our oxidative phosphorylation and chemiosmosis. This is where all of our electron carriers are headed. They will give their electrons ultimately to oxygen. In doing so, we will utilize the energy in the electrons to make ATP. This step is so effective, it is where the majority of ATP in the cell is going to be made. This step also requires oxygen. Let's start with glycolysis. Again, this happens in the cytosol of the cell. It's the initial step in oxidation of glucose. You will not be responsible for memorizing all of the structures in each step, but you will be required to understand what's going on. The overall reaction of glycolysis is to break down glucose from a 6-carbon molecule to 2 molecules of pyruvate. This does not require oxygen, so almost all organisms are capable of glycolysis. In order to do this, we have to begin with an energy investment phase. We have to put in some ATP to get the reaction started, just like lighting a match to start a bonfire. At the end of glycolysis, there's an energy payoff phase where you get to harvest some of the energy from this stage. Remember, glycolysis is a series of 10 different steps. You are seeing the first phase here, the energy investment phase. This has 5 steps to it. Keep track of the carbon molecules during this discussion. First thing that happens, glucose enters cytosol and is phosphorylated twice, here and here. The resulting molecule can be split into 2 3-carbon molecules. You don't need to memorize all of the intermediate molecules, but you need to know that this phase starts with glucose, and you have to add 2 ATP and end up with 2 3-carbon molecules. Notice that one of the molecules is called G3P, or glyceraldehyde 3-phosphate. We'll come back to that later when we talk about photosynthesis. The next phase is where we start to make some ATP, our energy payoff phase. For each of the 3-carbon molecules from the first phase, we can strip off 1 electron and reduce 1 molecule of NAD to NADH. Since we have 2 of the 3 carbon molecules, that means we get a net production of 2 NADH, 1 from each 3 carbon molecule. We also get to harvest 2 ATP here and here. But remember, we had to put in 2 ATP to light the fire under this reaction, so our net gain is only 2 ATP. Look at the product we get from our series of reactions. This is pyruvate. Remember, for each molecule of glucose that had 6 carbons, we get 2 molecules of pyruvate that has 3 carbons. This is the slide you want to remember. You start with glucose and have to add 2 ATP during the investment phase. During the energy payoff phase, you get 4 ATP produced and 2 NAD are reduced to NADH. Remember, these electron carriers are now full of electrons and hydrogens and will make their way to the mitochondria for use later. All of this happens in the cytosol of the cell, and you don't see any oxygen gas here, so you know that no oxygen is needed. Your end product, 2 molecules of pyruvate, will go on into the next step of cellular respiration, and the water produced is waste. Go ahead and pause here and talk yourself through what I just went over. And remember, glycolysis happens in the cytosol and does not require oxygen. The second phase of cellular respiration starts in the mitochondria. We now have 2 molecules of pyruvate for every molecule of glucose we started with. Pyruvate is a 3 carbon molecule. Pyruvate is transported into the matrix of the mitochondria, where it is oxidized. It loses electrons and a carbon molecule, which forms CO2. In doing so, a molecule of NAD is reduced to NADH. In this step, for each molecule of pyruvate, you produce 1 molecule of CO2 and reduce 1 molecule of NAD. The end product is acetyl-CoA. Since you had 2 molecules of pyruvate for every molecule of glucose, your net production in this phase is 2 NADH, 2 acetyl-CoA, and 2 molecules of CO2 waste. Now here's where it gets crazy. This is the citric acid cycle. We're still in the matrix of the mitochondria, and we have 2 molecules of acetyl-CoA that we created during pyruvate oxidation. This molecule still has 2 carbon and some oxygen and a lot of hydrogens left on it. Inside the matrix of the mitochondria, acetyl-CoA enters the citric acid cycle. This is where we finish breaking down what's left of our glucose molecule. Acetyl-CoA combines with a molecule called oxaloacetate. Oxaloacetate is a 4 carbon molecule, so once they combine, you now have a 6 carbon molecule. There are a few steps where the 6 carbon molecule is rearranged to make it easier to harvest electrons. It is now called isocitrate. The molecules are further rearranged and electrons and carbons continue to be stripped away, resulting in 3 molecules of NAD being reduced to NADH, 1 molecule of FAD being reduced to FADH2. The remaining 2 carbons are removed and combine with oxygen to form carbon dioxide waste. We also produce 1 molecule of ATP by substrate level phosphorylation. After we have stripped off anything remaining from our original glucose molecule, the resulting 4 carbon molecule is converted back into oxaloacetate through a series of reactions. So it's ready to be combined with new acetyl-CoA coming into the cycle. For each molecule of glucose, we get 2 molecules of pyruvate, which is oxidized into 2 molecules of acetyl-CoA. We go through the cycle twice, so our production per molecule of glucose looks like this. Pause here to write down the net products per molecule of pyruvate and the net products per molecule of glucose. This is what you're responsible for remembering. Oxidative phosphorylation is the 3rd and final step in cellular respiration. This is a process that happens across the inner mitochondrial membrane. There are 2 phases here. One is the electron transport chain, where we're going to be using the energy that is harvested from our electrons to pump hydrogen across the membrane, and chemiosmosis, where we're using the potential energy stored in the hydrogen gradient to create ATP. In eukaryotes, The electron transport chain is in the inner membrane of the mitochondria and consists of a series of 5 protein complexes. The electron carriers that we use to pick up all of our electron passengers stop here to drop off the electron. In the case of NADH, it stops at complex 1 and transfers its electrons, reducing complex 1 and oxidizing NADH back to NAD. Some of the energy is released, and the protein of complex 1 uses that energy to transport a hydrogen ion across the membrane. The electrons are transferred from complex 1 to the next protein, and then to complex 3. Again, the reduction of complex 3 releases energy, which is used to pump a proton across the inner mitochondrial membrane into the intermembrane space. The electrons are pulled along the chain as each complex is more electronegative. It exerts a stronger attractive force on the electrons than the last complex. In complex 4, the redox reaction again releases energy to pump a proton across the membrane. FADH2 skips complex 1. FADH2 skips complex 1 and releases its electrons, reducing complex 2. All of the electrons from both electron carriers end up at the end of the chain, where the final electron acceptor is oxygen, the most electronegative element. Oxygen receives the electron, is reduced, and binds to hydrogen, forming water. What we have done in this whole process is a transfer of stored energy in the covalent bonds of glucose to the potential energy stored in the electrons of the electron carriers, to finally converting that potential energy to energy stored in the gradient of hydrogen ions across the membrane. This large gradient we have set up is called the proton motive force, and we can harness it to do work. The hydrogen ions are charged ions, so they cannot diffuse back into the matrix. How do we use this to our advantage? The last slide we talked about was the electron transport chain. This is a process by which we set up our proton motive force, or our proton gradient. Here, chemiosmosis. This process also has a protein complex embedded in the inner mitochondrial membrane. It's called ATP synthase. ATP synthase makes ATP. It acts as a water wheel, allowing all of those hydrogen ions that are in the intermembrane space to flow through it, rotating the wheel. We're talking about the potential energy stored in the gradient turning into kinetic energy during the rotation of ATP synthase. ATP synthase then uses that kinetic energy to force a phosphate group onto ADP, storing the kinetic energy as ATP. Now, the energy is stored in a form that the cell can easily use. This form of making ATP is very efficient and gives you about 32 ATP per molecule of glucose. What happens to cellular respiration if no oxygen is available? Complex 4 is left holding its electrons. It doesn't have an oxygen there to pass them to, and that causes a backup at cytochrome C and complex 2. They are all holding onto their electrons. This causes a huge traffic jam as NADH and FADH show up ready to unload their electrons, and there is no available complex ready to take them. Pretty quickly, all of the available NAD and FAD have been reduced to NADH and FADH2, but they cannot unload their electrons. This causes a backup into the citric acid cycle, and the whole system stops. What about organisms that either never use or temporarily don't use oxygen? They can depend on glycolysis and the 2 ATP produced there to survive. However, you would still run into problems because you would run out of NAD without anything to oxidize it back from NADH. Organisms have evolved 2 mechanisms to deal with this problem. In the first, pyruvate is converted to lactate, or lactic acid, which results in the oxidation of NADH back to NAD. This is a strategy used by some microbes and also in your muscles. This can give you a small amount of time until you can start burning oxygen again, but you will not survive for long on anaerobic respiration. Other organisms can convert pyruvate to an intermediate, and then NADH is oxidized to NAD with a waste product of ethanol. Yeast do this process very efficiently, and we get to enjoy the product. That's it for today. See you in class.